The effects of vitamin D on the production of oxidative stress in C. elegans. In the past 100 years, there has been a dramatic increase of the average life expectancy of humans. In 1900, the average life expectancy of a male in the United States was 48 years of age. For a female, it was 51 years of age in the United States, according to the U.S. Census Bureau. In 2000, the average life expectancy of a male in the United States was 71 years of age. For a female, the life expectancy was 80 years of age, according to the U.S. Census Bureau. The most probable reason for this are the new advances in research on what increases life expectancy. Due to this research, conditions and medicine has improved to help increase the average life expectancy of human beings. There are many different theories for the reason and cause of aging. The most common theory is the mitochondrial free radical theory made by Fleming in 1978. This theory was first created by Fleming and is now the most popular theory for the reason of aging. This theory involves the free radicals that are created in the mitochondria due to oxidative damage. Oxidative damage is caused by an imbalance between the systematic manifestation of radioactive oxygen species, also known as ROS, and a biological system's ability to detoxify the ROS and to repair the damage made by the ROS. These free radicals accelerate the aging of the cell, which makes the organism grow older and lead to its eventual death. Oxidative damage is caused by an imbalance of ROS and antioxidants. There are many different ways to decrease the amount of oxidative damage on the body. The most common way is to increase the amount of antioxidants in an organism's diet. Antioxidants are created naturally by the body, but unfortunately, the amount of antioxidants made by the body are not enough to defend from the constant damaging effects of oxidative damage. Some foods that are high in antioxidants are blueberries, raspberries, strawberries, blackberries, apples, pecans, and cherries, which help defend from this damage. Some vitamins that have been proven to increase resistance to oxidative damage and help the breakdown of free radicals are vitamin C and vitamin E. There are many different chemicals in the body that create oxidative damage: hydrogen peroxide, superoxide, organic hydroperoxide, and many more. The most common one of these chemicals is hydrogen peroxide. Hydrogen peroxide can be measured by using a ROS assay. The amount of hydrogen peroxide in an organism has a direct correlation to the amount of oxidative stress in an organism. Vitamin D is a nutrient absorbed through the epidermis of an organism and then synthesized in the liver. Vitamin D can also be obtained by taking dietary supplements and in foods in our diet. Previously, low levels of vitamin D have been associated with Alzheimer's disease and has also been linked to the developments of different cancers and heart diseases. Vitamin D deficiency could also lead to the weakening in the body's immune system, which makes it susceptible to viral diseases such as influenza. It has also been proven that vitamin D deficiency can cause a decrease of bone density, which causes weakness. The most common type of vitamin D in humans is vitamin D3. In this experiment, C. elegans will be used as the model organism for a variety of reasons. Since C. elegans have a short lifespan, experiments can be done with them in a short duration of time. C. elegans also reproduce very frequently and in large amounts, which helps increase trial size, which will make the standard deviation more accurate. They would also be easier to examine throughout the experiment, and ROS assays can be done with them. Yi-Fen Li did an experiment on vitamin D3's ability to increase stress resistance in non-malignant human prostate epithelial cells. The vitamin D used in this experiment is 1,2,5-hydroxyl vitamin D3, the most potent and active form of vitamin D in the human body. Normal metabolisms create oxidative stress, which causes free radical decay, which harms the organism's DNA. The free radical decay has a large role in aging. In this experiment, 4 different strains of human prostate epithelial cells were used: BPH1, RWPE1, CWR22R, and DU145. In PH1 and WRPE1, there was a clear increase of oxidative stress resistance in the 100 nanometer D3 groups compared to the control groups. In the CWR22R and DU145 groups, there was no significant difference between the two groups. Messing did an experiment on the effects of vitamin D3 on the lifespan of C. elegans. She had a control group, 0.001 microgram group, zero point 0.1 microgram group, 1 microgram group, 10 microgram group, 100 microgram group, and 1000 microgram group. All of these groups showed an increase of average lifespan of C. elegans except the 0.1 microgram group. The only group to show a significant increase in lifespan compared to the control group was the 1000 microgram group. According to Lee's and Messing's experiments, it would be expected that the 100 microgram group, 50 microgram group, 100 microgram group, 500 microgram group, and 1000 microgram group in this experiment will all show an increase of average lifespan of C. elegans compared to the control groups. The purpose of this experiment is to see if increased amounts of vitamin D will help decrease the production of oxidative stress in C. elegans. In this experiment, there will be 6 different groups: the control group, 10 microgram group of vitamin D3, 50 microgram group of vitamin D3, 100 microgram group of vitamin D3, 500 microgram group of vitamin D3, and 1000 microgram group of vitamin D3. There will be 5 trials done with each group. In each trial, 20 C. elegans will be used per trial. These C. elegans will be from the wild type strain. The E. coli fed to the C. elegans will have the OP50 strain. To measure the amount of hydrogen peroxide each C. elegans produces, the Amplex red hydrogen peroxide assay shall be used. Once prepared, the C. elegans will emit a fluorescent light, which wavelength will be measured in nanometers by a spectrophotometer. The C. elegans in each group will be the same age. This will be accomplished using the age synchronization protocol. Using the Amplex red hydrogen peroxide assay, the amount of hydrogen peroxide produced by each C. elegan can be determined. As the fluorescence of the C. elegan increases, the amount of hydrogen peroxide produced by the C. elegans is higher. The Amplex red hydrogen peroxide assay will be purchased from Life Technologies. The spectrophotometer needed from this experiment is already found in the lab. The cuvettes needed to use the spectrophotometer will be bought from Carolina Biological. The C. elegans will be purchased from CGC and will be the wild type strain. The C. elegans will be fed E. coli OP50 bought from CGC. The C. elegans will be cultured in C. elegan agar bought from Carolina Biological. The vitamin D will be bought from Fisher Scientific. A micropipette will be bought from Carolina Biological to make sure all the measurements of vitamin D put into each group is accurate.